Mergene1000 Cell-Specific mRNA Transfection Reagents Operation Guide. ProCell Mergene1000 mRNA Transfection Reagents are specifically optimized for delivering exogenous mRNA into designated cell lines, offering high transfection efficiency, low cytotoxicity, and excellent stability. This video demonstrates the transfection workflow using the Mergene1000 Hep G2 Cell-Specific mRNA Transfection Reagent as an example. Cell seeding. 1 day before transfection, dissociate healthy Hep G2 cells and seed them into a new culture vessel. In this demonstration, a 6 cm culture dish is used. Add an appropriate volume of pre-warmed Hep G2 cell-specific culture medium to the 6 cm culture dish. Prepare a single-cell suspension of Hep G2 cells and determine the cell count. Seed 1,500,000 cells into the 6 cm dish with a final culture volume of 5 ml. Gently move the dish in a figure-8 pattern to ensure even cell distribution. Incubate the culture dish at 37 degrees Celsius in a 5% carbon dioxide incubator for 24 hours. Before transfection, observe the cells under a microscope. A confluency of 70 to 90% is optimal. The culture period may be adjusted according to actual cell growth and confluency. For other culture vessel formats, refer to the recommended seeding densities in the table or calculate based on the culture surface area. Preparation of transfection complexes. Prepare 1 sterile centrifuge tube. Add 10 microliters of Mergene1000 Hep G2 Cell-Specific mRNA Transfection Reagent and 490 microliters MEM medium supplemented with NEAA. Mix gently by pipetting. Add 2 micrograms of sterile mRNA to the diluted transfection reagent and mix gently by pipetting. Incubate the transfection complex at room temperature for 5 to 10 minutes. For other culture vessel formats, refer to the recommended transfection complex preparation volumes in the table or calculate based on the culture surface area. The amounts listed above are the preparation quantities required per well. Please calculate the total preparation volume needed according to the specific experimental conditions and requirements. Cell transfection and observation. Add the prepared transfection complexes dropwise and evenly across the 6 cm culture dish. Gently swirl the dish in a figure-8 pattern to ensure uniform distribution. Incubate the culture dish at 37 degrees Celsius in a 5% carbon dioxide incubator. Gene expression can be analyzed after 12 to 24 hours of incubation.